Fruit flies, a common summer nuisance. But did you know that about 60% of their genes are similar to humans? In October 2024, an international research team at Princeton University's Neuroscience Institute successfully completed a detailed neural map, also known as a connectome, of the adult fruit fly brain. Their findings were published in 9 research papers in the prestigious journal Nature. The fruit fly is a tiny insect with a brain smaller than 1 millimeter. Since brain function is determined by neuronal activity in synapses, understanding how neurons are connected can provide key insights into how the brains of humans and other organisms work. That's why scientists from 12 countries, including the United States, the UK, Israel, Germany, South Korea, and the Philippines, joined forces to map the fruit fly brain. As a result, they discovered that the adult fruit fly brain contains 140,000 neurons and 54.5 million synapses. While past studies had mapped the brain structures of C. elegans and even fruit fly larvae, this is the first time such a complex animal has been mapped at the adult stage. The brain consists of neurons, which are nerve cells, and synapses, which connect them. A synapse is the gap between neurons where neurotransmitters are exchanged. For nearly a century, scientists have tried to map these intricate neural connections, much like an intricate web. In the early 1900s, Italian anatomist Camillo Golgi used staining techniques to visualize neurons under a microscope. He then painstakingly hand-drew what he observed, creating detailed illustrations of neural structures in mice, the hippocampus, and other brain tissues. His work led to the discovery that neurons are the fundamental building blocks of the brain, earning him the 1906 Nobel Prize in Physiology or Medicine. Today, 2 major challenges must be overcome to create a complete brain map: identifying every single neuron in the brain, mapping all the synaptic connections between these neurons. In the past, microscopes alone were not advanced enough to tackle this challenge. But with the development of electron microscopes, which can observe extremely small structures at high resolution, scientists could finally meet these 2 conditions. However, there was still one major obstacle. Mapping millions of neurons by hand would take an enormous amount of time and effort. This is where AI came in. Using artificial intelligence, researchers reconstructed the entire adult fruit fly brain in 3D. In just 6 years, they successfully mapped every neuron responsible for receiving and sending signals, completing the first ever full fruit fly connectome. Without AI, it would have taken a single person 50,000 years to accomplish this task. This is a groundbreaking achievement made possible by the fusion of neuroscience and computer science. But why did scientists work so hard to complete the fruit fly connectome? Because in the near future, this technology will allow us to map the brains of larger animals, and eventually, the human brain itself. If we can achieve that, we might finally uncover the causes of brain diseases such as Alzheimer's and Parkinson's, bringing us closer to potential cures.